So I realized that I stand between you and lunch, so I'll get to it. So in our group, we spend a lot of time thinking about how interactions at the submolecular level, so thinking about residue level interactions, contribute to collective behaviors of biomolecules and contribute to mesoscale condensate properties. I had to sprinkle in some mesoscale for you, Stephanie. And so one of the questions that we've become very interested in is what drives small molecule partitioning into condensates. So if you have a condensate, for example, and you have a set of small molecules in the surrounding environment, what drives some molecules in and keeps others out? And I think that this question is very relevant because it allows us to understand condensates in terms of as microenvironments, and I think of them a lot as these fluids, these solutions that have their own environment that is distinct from everything else around them. But also helps us in understanding function, particularly if there are cases where we see particular cofactors or metabolites are sequestered into various condensates that can give us some clues about activity, for instance. And one particular area that we've been interested in is in the idea of designing drugs that can target biomolecular condensates and how understanding this interaction between small molecules and condensates can help us in that pursuit. Another aspect involves engineering condensates for particular functions. For example, in designing metabolic condensates that can drive chemical synthesis, for instance, understanding these interactions between small molecules and condensates is very important. And so what are some of the factors that could possibly govern whether small molecules localize to biomolecular condensates? We've gotten a lot of clues from recent work. I highlight some of these papers here, and some ideas that have been put forward is that the properties of the small molecules are very important. Thinking about the hydrophobicity, the size, the charge, the flexibility, and so on of the molecules. Another idea is that the properties of the condensates is the determinant factor. So thinking about the molecular composition of the condensates, the pore size, the water content, basically the environment that the condensate makes selects what comes in and what does not. And they're less prevalent, but I think other important ideas is binding interactions between small molecules and macromolecules that are within condensates. We heard a lot of examples about like enzymes being in condensates in these talks and in this meeting, and these enzymes have pockets that can bind, and we have other macromolecules that can exhibit specific interactions with small molecules. And so we wanted to take on this question in our group to be able to understand what's going on and to give a submolecular picture of what could be the determinant factors. And so this work is really driven by Alina, who is a postdoc in the group, and was done in collaboration with Pablo, who is also here at this meeting. Sorry to put you on the spot, Pablo. And Danielle, who is this talented undergraduate student that just graduated and will be going on to do his PhD. And so we wanted to take this simulation-driven approach to understand what factors may be at play here. And so the first thing we decided, okay, let's select which small molecules that we're going to look at. And so let me define what I mean by small molecules for the context of this talk. I'm talking about molecules that have roughly a molecular diameter of around 6 angstroms, which is roughly the order of a size of a single amino acid. And we're selecting compounds that are totally uncharged, so we don't want anything with charges in them yet. And we want a sizable variation in hydrophobicity of the molecules. So if we look at log P, which characterizes the partitioning between octanol and water, that gives us an estimate of the hydrophobicity of the compounds. And so we choose a range that roughly spans the range of small bioactive molecules. And so we came up with this set of, by selecting for these criteria, we came up with this set of about 17 compounds that we're going to be looking at. And so our approach of or one might say, okay, the gold standard approach that we would want to look at this problem is by using atomistic simulations. And these simulations are very powerful because they allow us to really have detailed information about interactions between small molecules and condensates. But you heard on Monday from Ben Salk that these simulations are extremely costly. Just to give you an idea, if I have a protein that has about 500 residues, and I take 64 copies of this protein and I put it in an atomistic simulation and I want to study its phase behavior, this boils up to about the order of 1 million particles. Takes about a month to just get 1 microsecond of sampling. So if we want to really search this sequence space in a quote unquote high throughput manner, screening these interactions at this level, all the resources that we have will have to go towards this one project, and we have other projects. And so, what are we going to do? Do we not do atomistic simulations? Well, our idea was that, okay, we can still take an atomistic approach, but let us reconfigure how we're going about this problem. And so the idea is that we still want to do atomistic simulations. So we have explicit solvent and ions. We're representing our proteins and water and ions. This is just some force field information for the simulation people in the room. We're simulating the systems in the NVT ensemble. And the key idea here is that instead of looking at all of the proteins that you've seen in this meeting, we're going to take a simple sort of model peptides that can condense at 300 Kelvin, right? And so in work by people like David DeSancho, they showed that if you take peptides that are about of the order of 6 amino acids long, these things can phase separate and they can condense. And so we're going to say, let us take these model systems that can condense at 300 Kelvin at 150 millimolar salts, and let us use these as sort of model environments of what type of environments that condensates may make. And so what we want to do is we want to vary the composition of these peptides so they can mimic different condensate environments. And so what we're doing here is that we selected these 4 systems where if you go from this sequence to that one, we have increasing hydrophobicity of the sequences, and so we go from being environments, condensed environments that are highly polar or enriching polar residues to environments that are enriched in your aromatic and aliphatic residues. And what we're going to do is that we're going to take the systems and we're going to add our small molecules to them, where we're in each system we're going to add one of the different 7 small molecules that we have identified. And from this sort of model condensate systems, we want to see what we can learn. So in total, we have roughly about 65 systems that we're looking at. And so what can we learn? Well, one of the first things that jumped out at us, which was not surprising, because this result was postulated in the literature already, that if we measure the partitioning coefficient of the small molecules in the systems, so here I am defining my partition coefficient as the number of molecules that are in the condensed region versus the number of molecules that are in the dilute phase, we see that as we vary the hydrophobicity of the small molecules, we have a very strong dependence of this property on partitioning. So as molecules become more hydrophobic, they partition more readily into the condensates. And so because we have these varied environments that we're looking at, we can ask how this relationship changed from one environment to another. So if I swap out this peptide for this one, and I look at the same set of small molecules, and I look at how the partitioning varies as a function of the small molecule hydrophobicity, we see here that we have a less strong dependence of the small molecule hydrophobicity on partitioning, right? So this nice, relatively strong dependence that we get here is weakened when the environment that the molecules are partitioning into become more polar. But we can take it a step further because we have this set of 65 systems, and we can kind of zoom out and see what is the big picture here. Well, what I'm doing here is I'm taking my 4 different environments. So these are the 4 different peptide-based condensates that we have. And for all of the small molecules that we have, we're plotting the partitioning coefficient as a function of the environment. And we see here that if we go on the side where we have increasing hydrophobicity, we get these environments exhibit a very strong selectivity for the molecules that for molecules that partition into them, where if we go along this side where we have a very more sort of like polar environment, they seem to not care about the small molecules that we put into the system. They relatively don't partition into these types of systems. So condensates can differ particularly for the model condensates that we look at, they can differ significantly in their partitioning landscape. But another thing that we observed is that if you look at the slopes of these lines, they change from one small molecule to the next, right? And so it's not just the condensates that are selecting for the small molecules, but there is some sort of sensitivity that the small molecules have to the condensate environment. And we can quantify that. So if we take these slopes and we quantify that, we call this partitioning sensitivity. If we plot that against the log P of the compounds, we find that high log P compounds tend to have a greater partitioning sensitivity. So variation in partitioning is heterogeneous among the compounds. Some show strong variability, others do not. And so we started to think about this problem and think about the idea, well, the first thing that we wanted to do, we're very excited about thinking about drugs and how you can target these types of systems. And if you think about a lot of the examples that we've looked at, the fraction of polar residues in things like FUS and HNR and PA1 is very high. So these systems have a high fraction of polar residues. And so our data is telling us that condensates that are enriched in polar residues show poor selectivity. And so how do you get around targeting these types of systems if they are enriching the residues that diminish this sort of selective effect? And so we went back and we looked at our data again, and we're asking ourselves, can these condensates that are enriched in polar residues exhibit any sort of selectivity? And what jumped out at us is that if you look at the data, you see that relatively your correlation is very weak with log P, but you do see some examples of some compounds that partition one is basically not that high in this case, but that show preference for the condensate over the dilute phase. We thought that should not be the case because, like, from an entropic standpoint, we're thinking, if there's no sort of like favorable interactions that are driving these things inside, if the environment is very similar, why would they show any sort of preference for the condensed environment? And thanks to some good feedback that we got from the reviewers, we went back and we looked at this data again, and what we noticed is that for these compounds that exhibit selectivity into these polar enriched condensates, these compounds have a high dipole moment. And so if we look at if we take our data and we project it again, looking at the dipole moment of these compounds versus their partitioning as a function of the environment that they partition into, we find that highly polar condensates can exhibit selectivity for high dipole moment compounds. And so that points us to ideas that even in environments where you are enriched in these polar residues, condensates can exhibit a selectivity, or at least selectivity can be engineered in these types of systems. So one idea that we have is that this dipole-dipole as well as dipole-pi interactions can help to override the effects of hydrophobicity in these types of systems. What I've told you so far is all well and good. We've been looking at these model systems, simple systems, and using that as a way to begin to understand sort of what are the physical chemical factors that drive partitioning in these types of systems. But we really want to model things like fast, right? We really want to model large condensates made up of many different types of components and study these types of systems. So how do we go towards larger length scales? Well, one of the things that we can do is that we know that our atomistic simulations are very costly. We can adopt a coarse graining approach, right? So instead of representing all of our amino acids explicitly in our system, we can coarse grain them. So we take each amino acid, for instance, and we map it onto a single interaction site. And using a coarse graining approach, we can essentially accelerate the rate at which that we can study these small molecules. The caveat here is that how do we go about getting parameters for these small molecules that capture the essential physics that is driving the partitioning? And so I won't give you the longest spiel that I was going to give you, but the shortest spiel is that we essentially going to start from a place where we have designed, in my previous work, we've designed this coarse-grained approach for predicting phase behavior of disordered proteins, and we showed in previous work that this model is able to predict well both single chain and collective phase behaviors of disordered proteins. And so we're using this as a starting point to start asking what are the essential ingredients that we need to add to be able to predict partitioning well for these small molecule systems, which is complementary to the work that we heard earlier. And so one of the things that we can do is we can think about what interactions these small molecules make with themselves. And to do that, to capture these effects, we use this technique we call umbrella sampling, where we can compute a free energy of interaction between small molecules. And our first foray at this, we were like, oh, we're just going to take a brute force approach to be able to optimize the small molecule parameters. So the high level is that we have a coarse-grained model of the proteins, and we want to figure out what interactions that we need between the proteins and the small molecules. And so we're going to take a brute force approach where we have all of these atomistic simulations, and we're going to optimize the parameters of our small molecules so that they can recapitulate what we see in the all atom systems. And the short end of it is that if you do this brute force approach, you get good predictability, you get transferability, but this is not practical. This is not practical. It's extremely labor intensive. For every small molecule that we want to study, we have to do at least 40 different types of systems, multiple rungs of optimization to get around that. And so how can we develop a sort of more intuitive way of going about this? Well, essentially what we want to do is we want to figure out what is the effective interaction that a small molecule makes with a condensate makes with a protein in a condensed environment. And so we define some sort of effective interaction where we're saying, based on our atomistic simulations, our atomistic simulation told us that both the properties of the small molecule as well as the properties of the condensate matter. And so in this sort of effective interaction that we're designing, we're taking into account the affinity that small molecules have for proteins. And here we use this ensemble approach where we simulate proteins with small molecules, and that gives us an approximate sort of interactions between the small molecules and different protein residues. This is essentially our way of capturing a sort of chemical fingerprint for the small molecule. And we also take into account the environment that the condensate, that a given residue, or the impact that a given residue has on the condensed environment. And the short end of that is that by taking into account both the interactions that the small molecules make with residues, as well as thinking about the condensate environment, this empirical relationship that we come for fitting the parameters for the small molecules does very well in comparison to our brute force approach. And so we want to now see like how well we can actually predict partitioning, and can we do this for much larger systems. So first I just show you briefly what we get if we look at our model condensates. So I showed you a bunch of data for model condensates in all atom, and we see that if we do relatively well if we compare for our model systems that you've seen before. If we select only for the hydrophobic condensates, we also do relatively well. But what we find is that when condensates are enriched in more polar residues, we have a systematic overestimation of the partitioning there. And so that is telling us that this empirical relationship that we derive is neglecting some effects of the solvent that we're just not capturing very well. However, we say, okay, we still want to understand if this thing is even transferable, because right now we just have the short peptides, that the environment that short peptides make will be very distinct for what you can make if you have a very long polymer. And so we're going to select the compounds here that we predict the best using the coarse-grained approach. Well, not all of them, but a subset of them. And we're going to say, can we predict partitioning into biologically relevant condensates? And to do that, we take our favorite condensates, which is the low complexity domain of FAS. What we do, we use this technique where we simulate force LCD condensates in the coarse-grained model, and then we use this back mapping approach to map this condensate back to the atomistic length scale, and we add small molecules to the system. So here we're going to do this month-long simulations that I talked about earlier to study partitioning into the system, and we're also going to do these simulations, which takes of the order of less than a few days, right? And we're going to compare to see if we can even capture these effects. So the results that I'm going to show you on the next slide, we'll only do this for 4 compounds because this is very expensive. But we see that if we look at the partitioning that we get using a sort of a coarse-grained, a minimal approach versus this long time scale atomistic simulations does fairly well. We do still see overestimation of partitioning using the coarse-grained approach, and again we think that is because there are some solvent effects that we're neglecting or we're not accounting for well, but this gives us some promise that this sort of like minimal approach can help in more efficient screening of small molecule interactions with condensates. We also see in our simulations that if we take a given small molecule and we track it over the course of the entire simulation, and you ask which protein residues does that small molecule interact with, do we get the same sort of interaction landscape in the coarse-grained approach that we get in the atomistic simulations? We see good agreement there, but as you can see, these frequencies are much lower in the coarse-grained simulations than in the atomistic simulations, and we think that is because we have really smoothened out the landscape, and so this sort of like specific interactions that you will get in the atomistic simulations, we lose them when we go to this level. And so I will stop here for now and say that our simulation so far seems to suggest to us that partitioning is influenced by both the properties of the small molecule and the environment, that hydrophobic compounds partition more strongly, with hydrophobicity enhancing sensitivity to condensates, and that condensates can vary in their selectivity. And an important point that I don't think that I made here is that we do have systems where we can, such as in condensates enriching polar residues, where they can select based on the dipole moments of compounds, and from a coarse grain and from a more simulation point of view, we think that in order to accurately predict this partitioning, you must account for this small molecule properties as well as the complex environments of the condensate. Moving forward, we want to incorporate more factors that may be important. So increase the complexity of the small molecules, so we didn't use anything with charge. Increase the sizes and see how well, as flexibility of molecules come into play more, how well we can predict these behaviors, as well as thinking about the complexity of the condensate itself. For this set of work, first LCD was like the gold standard in terms of like how big that we could go, but we really want to start thinking about multi-domain proteins, systems with RNA, and thinking about how these specific interactions might be important. And we try to capture some of these ideas where specific binding interactions. I really like the talk earlier with CD code, Agnes talk, where you see a lot of these structured domains are very important in the biomolecular condensate, and we think that that is one idea that has been left out in the discussion. And I will end here now. Thank you for your attention, and sorry for keeping you from lunch.
Very nice, Jerelle. So what it seems like you made early choices to look at these small compounds that are uncharged, but some of your conclusions seem to be about like whether or not small molecules sort of in general can be partitioned into condensates in general. So how much do you think it matters that the molecules themselves are going to be different, and so these things really, the conclusions apply very much to small nonpolar compounds, but maybe there's just the charge itself is the reason why the nonpolar things are not partitioning in the way they
yeah, I think charge is going to come into play a lot, especially if you think about things like RNA. Many proteins that form condensates have net charges, have a lot of charge residues along their lengths. I think one of the things that we pulled from like when we started this project 2 years ago was thinking about sort of like the discourse that was going on in terms of like what is important and what is not. And as your comment really highlights, what is important and what is not is very much determined on like what you're looking at, right? And you see cases where people say, yeah, the condensate environment does matter, where other conclusions get at the small molecule characteristics is the key determinant. But if you look at sort of like what the data sets are, there is like very difference in terms of the data set. So as we increase the complexity of the systems, I think the conclusions might likely shift, and we might start seeing more interesting behaviors.
Yeah, I had the same question as Alan. And so in terms of how you chose the first 17 compounds, there's some kind of rule about like small molecules to be drugs have to have like different criteria. So was the rationale for choosing uncharged things, are those better drugs in general?
No, we didn't think about drugs.
Okay.
So we just thought about like small organic compounds to look at. The long end of this question is that we originally thought that we were just going to go for the drugs that are like super important, that have been shown to be important for targeting condensate. Turns out it's very expensive and difficult to sample some of these large systems. Okay. And so our decision was, can we understand sort of what are the physics and chemistries that are involved for simpler systems, and use that as a way of thinking about what are the essential parameters that we need in these types of models. But yeah, we did not necessarily, some of these molecules that we have will kill you.
Okay. Yeah. No, cool. So just to be really specific, so the take home that hydrophobic condensates are more selective than polar condensates, that is specific to this set of molecules, and that like correlation might be totally different for a different set of small molecules. Is that true, or am I missing the point?
I think that we might find different conclusions as we increase the complexity.
Cool, thanks.
Yeah.
Hi, Jerelle. Do you have a way to see whether the small molecules that enter change properties of the condensate themselves through your simulations?
Yes, so that is something that we're looking at right now. That is something that we're looking at right now. And one of the ideas that we have around that is that perhaps the partitioning is not the important thing that you need, and that, yeah, we have a lot of ideas around what different effects that you can have, and what properties of small molecules may be important, and perhaps it's not partitioning.
Hi. Do you see in your simulations the compounds actually dissolving the condensates, or just partitioning?
So we had some earlier simulations where we saw somewhat dissolution, but I think like especially the ones that we select in this set, we did not see any sort of like strong effect.
Because there are some drugs, right, that are known to do that, so it could be a good control to see that in your
So now
simulation.
we're looking at these molecules and hope to share something about that.
Cool.
Last question before lunch, and then maybe you can discuss with Jerelle later.
Yeah, thank you very much for this interesting talk. I have a comment and a question. So I'm not really surprised by the deviations between the atomistic and the coarse-grained simulations, because when you parameterize the coarse-grained interactions, you always get a free energy. It's not an energy. And so you have entropy contributions to that energy function on the coarse-grained level, and that means you are, by very principle, you have a state point dependent interaction. And this is something one, I think, has to take into account also when one looks at such deviations. And I have a question concerning your coarse-grained simulation itself. Since you there work with an implicit solvent type of model, and your particles are fairly dilute outside the condensate, did you run a grand canonical simulation there that you can add new particles so that you can avoid finite size effects?
No, we did not do grand canonical ensemble in the simulations, no.
Yeah, thank you.
The only criteria that we had in terms of like we just try to match the concentrations that we had in the whole atom. Yeah.
Okay, let's thank our speakers again.